Okay, so guys, today we are going to start this topic of speciation. In fact, I'm just going to wrap up a sort of recap from what we did last time. So, as the name indicates, it is the evolution of one or more species which arises from previously existing species. And as I told you in the last class also, that first I will discuss speciation under the topic of intraspecific, which means speciation or the evolution of one or more new species from previously existing species within one specific species, right? And then after we have done intraspecific speciation, we will be covering interspecific, which is common in flowering plants. And when I was teaching you polyploidy, in which we were doing a cross between two different species or distinct species of wheat plants, so we were actually doing interspecific speciation or interspecific hybridization. In other words, it would be not wrong to sum up that interspecific hybridization can lead to interspecific speciation. And again, this kind of speciation is not common in or practically not found in animals, but it is common in flowering plants. Why flowering plants? Because it is in flowering plants only that sexual reproduction or involvement of gametes takes place. Speciation can be intraspecific. So let's start with intraspecific. In intraspecific, we have allopatric speciation and sympatric speciation. Allo means other and patria is the word coming from land. So this means a speciation that happens within one species, but that one within one species has been geographically separated by some natural disaster, river, earthquake, whatever. And then this kind of speciation takes place. In sympatric speciation, the population of the organisms that belongs to the same species will occupy the same geographical area. Now, the common thing between allopatric and sympatric speciation is that both of them are classifications of intraspecific speciation, right? And this diagram explains very well how these two kinds of speciations will take place. You have a same species, which means intraspecific. Now, this species has been separated geographically like this, okay? And they have been separated for a long time. So after time passes, they sort of get some new characteristics, right? Which makes them a new species. And then this is sympatric speciation, in which you can see this one, that it's the same species. It has not been geographically isolated, but due to some other factors, some of the members of that species will develop different characteristics. Okay, those can be several factors. Okay, so now we move on to some details about these speciations. As the description says over here, the sympatric speciation, which means this one is actually the speciation that occurs even though the groups that have shown different characteristics, they are occupying the same geographical area, right? I told you this as well, that whenever you are describing speciation, you will describe the underlying points. In order for any population to evolve or to develop some new characteristics, it is a must that certain conditions should be fulfilled, in which you have gene flow. It should be interrupted. Because of interruption of gene flow, of course, a subpopulation becomes genetically isolated, which means I would say, in order to explain this gene flow or interruption of gene flow over here in speciation, I would say that when these two populations were not geographically separated, there was gene flow from this to this, right? But because of a geographical reason, now the gene flow between this population and this population has been interrupted, right? So because of that, the subpopulations become genetically isolated, right? Allele and genotype frequencies are changed. Natural selection is the process along with the effects of mutations and sexual recombinations. Genetic isolation for a long time will not allow this subpopulation to breed with its ancestor, and it becomes a new species. And one last important factor is the reduction of selection pressure on this subpopulation, which allows it to adapt to the environment and survive. Okay? For example, if we take into account this kind of speciation, I would say that initially all these trees were living together. A river separated them. Because of that, the gene flow was interrupted. There was no gene flow. The allele and the genotype frequencies changed if this goes on for a very long time. Random mutations will take place. Okay. Since they are geographically separated, the selection pressures acting on this population will be different, whereas the selection pressures acting on this population will be entirely different. So since the pressures will be different, the characteristics that they develop will be different over a period of time. New recombinations, allele recombinations or sexual recombinations will form, and the possibility of the evolution of a species which has some different characteristics will increase, right? So these are also all the factors that are involved in intraspecific speciation. Bottom line for all kinds of speciation is that it will only occur as a result of formation of barriers, which leads to the reproductive isolation between members of the population. Okay? And these are known as isolating mechanisms, which means that when speciation is occurring, there has to be some mechanism that isolates those two subpopulations. ठीक है? उसको isolate इस तरह कर दिया है कि उनके दरमियान gene flow नहीं हो सकता. So what are those isolating mechanisms? Let's just study those mechanisms. ठीक है? In fact, we can say those isolating mechanisms are actually barriers that do not allow the gene flow between two subpopulations. These isolating mechanisms, to an extent, have been explained in your textbook also. Usme I think paragraph ke form mein explain kiya hua hai. But this is just a table that gives you a very quick idea about how these different isolating mechanisms they operate. ठीक है, अब आपको इसको learn नहीं करना है. You just go through it thoroughly so that you have an idea कि कितनी तरह के isolating mechanisms हो सकते हैं. And in exams, any different example can be given to you and you will be asked to comment on it or talk about it. So generally we can say that these isolating mechanisms can be divided into pre-zygotic and post-zygotic, meaning that those that occur before the zygote is formed and those that occur after the zygote is formed. For example, case mein unhone example di hui hai over here. There is a temporal mechanism which occurs when two species mate at different times of the year. Okay? Frogs live in the same pond but breed during different seasons. So this means this kind of a reproductive isolation is temporal isolation. कि temporal का मतलब time से है ये कि you have sort of many frogs living in the same pond, but why do they don't mate? Because they are breeding, ठीक है, during different seasons, or maybe laying down eggs during different seasons. So that's why they don't mate. ठीक है? Ecological isolation might be कि ऐसी isolation जो कि जिसका cause ecological हो, which means it can occur when two species occupy different habitats. ठीक है? Lions and tigers can potentially interbreed, but they usually occupy different habitats. So that's what makes them isolated genetically. Behavioral species occurs when two species have different courtship behaviors, which means that in animals, you know, the male animal is attracted or attracts the female animal by certain kinds of behaviors, right? So it can happen that when there are two birds, but they are not mating with each other because their behavior is sort of alien to the other bird. So that's why, as it is written over here, certain groups of birds will only respond to specific species, specific mating calls. And let's say if the mating call for this bird is not, I can say, is not attracting this bird, right? So then this means these two birds will not mate with each other. Mechanical isolation, mechanical genetic isolation occurs when physical differences prevent pollination, right? Or prevent mating. This is very, very obvious that there is a certain breeds of dog are morphologically, structurally different. and that is why they are incapable of mating due to size. These are pre-zygotic isolating mechanism. They can be post-zygotic also, right? In post-zygotic, we have hybrid inviability, meaning that hybrids are produced, but they fail to develop due to reproductive maturity. Like certain types of frogs form hybrid tadpoles that die before they can become a frog. For example, that maybe two different species of frogs are mating, and they are producing some tadpoles or baby frogs, but since they are hybrid frogs, so they will die. And as I told you, this kind of interspecific hybridization is not compatible or viable in animals. So animals में usually इनके hybrids मर जाते हैं. Hybrid infertility means hybrids fail to produce gametes. जैसे भी हमने बात किया था ना कि when we were doing polyploidy, we said that the F1 hybrids are produced, but they are sterile. Same example that when we mate a horse and a donkey, we produce a mule. So mules are sterile. Mules are sterile hybrids resulting from a mating between a horse and a donkey. Okay? And hybrid breakdown means that F1 hybrids are fertile, but F2 generation fails to develop properly. Okay, so ऐसे भी हो सकता है. You're not supposed to remember each and every example. But this slide has been shared with you just to give you an idea that how these different isolating mechanisms can happen and how they can result in genetic isolation between two subpopulations so that their gene flow is interrupted. अच्छा. Details of allopatric speciation. थोड़ा बहुत पहले से ही पता है, थोड़ा सा और कर लेते हैं कि the word allo means other. Patria means native land. So speciation, that is characterized by the occurrence of spatial separation. Spatial separation का मतलब ये है कि species or populations separated by space. जाहिर है वो geographically isolated हैं, so they are separated by space, and that is known as spatial separation. ठीक है? We all know this. Geographical barriers, mountains, rivers, seas, habitat, separating two subpopulations, causing an interruption to gene flow so that the organisms are unable to mate, leading to reproductive isolation. Okay? And then adaptations to new conditions or genetic drift will change the allele and genotype frequencies. I think last time Anas was asking me कि genetic drift हो जाएगी इसमें. हाँ, sort of genetic drift तो हो रही है ना over a period of time. So it will change allele and genotype frequencies. Prolonged separation will lead to a new species evolving. ठीक है? Allopatric speciation. The example of Darwin's finches, in which Darwin is going to Galapagos Island to study the different kinds of finches, their beak sizes and beak shape. So he saw that these finches or these birds, they move from mainland to Galapagos Island. Okay? Now, when they moved from mainland to Galapagos Island, there was no competition present from endemic species. Okay? So the selection pressure was reduced. So slowly and gradually, this allopatric speciation started to occur, in which what Darwin saw was that he saw that the kinds of the finches and their beak sizes and shape were different on the Galapagos Island as compared to the mainland, because of course, these two islands differed in the kind of the nuts and the fruits, and the consistency and the shapes and the sizes of the fruits were present on these two different islands, caused the beaks of these birds to become different over a period of time. So this is because that those finches initially were living in on mainland, and then they migrated to maybe Galapagos, and that is why the difference in the shapes and the size of beaks occurred. So this kind of changes, these kind of changes that happen in the shapes and sizes of beak when these finches move from mainland to Galapagos Island will come under the classification of allopatric speciation, or isko hum adaptive radiation hi kehte hain. Why have I used this word? Because if you come across this in your exams, you should remember that there is no difference between adaptive radiation and allopatric speciation, right? Speciation that happens when two subpopulations are separated geographically. Picture of adaptive radiation in Galapagos Island, different kinds of finches, different shapes and sizes of their beaks because they are feeding on different prey. ठीक है? Sympatric speciation. Sympatric speciation, sim का मतलब same, patria means land. So speciation occurring in a population within the same geographical area. Genetic differences may accumulate allopatrically, which have been isolated for a much more shorter period of time. If these populations were brought together, hybrids may form where these two populations overlap. Acha. These hybrids have reduced fertility and serve as an efficient reproductive barrier to gene flow between populations of carrion and hooded crows. In time, selection pressure against the crossbreeding may occur, leading to speciation. Since it is occurring in the same geographical area, it is known as sympatric speciation. ये आप सबको बहुत confuse कर रहा होगा. In order to make it easy for you, ये बताना ये चाह रहे हैं कि initially a species was separated geographically. ठीक है? Separate होने के बाद अब ये दोबारा से आ जाती है. Any sort of this species that was separated allopatrically now comes to live with the same parent species, the ancestor species. तो अब ये एक जगह जमा हो जाते हैं, and now the crossbreeding between them becomes impossible because there is a reproductive barrier between these. ठीक है? Between the gene flow between these populations. So अब अगर जो है इनके दरमियान gene flow नहीं हो सकता, although they are living in a same geographical area, so we say that, and if this continues to happen for a long time, so we say sympatric speciation has developed. इसमें examples आपको different तरह की दी हुई हैं कि sort of over here they are showing you the original population and then the ecological isolation and reproductive isolation and then genetic divergence, जिसमें आपको एक ही population दिखाई है within one geographical area. This is the original population. ठीक है? अब इनकी ecological isolation हो गई है, right? Due to some reason, they are living in the same area, but there is ecological isolation that without some specific reason, they choose to live in another habitat, उसी area में, but थोड़ा सा different habitat. So they might become ecologically isolated. अच्छा, when they become ecologically isolated for a long time, and if this keeps on happening and happening and happening, they become a little different. You can see genetic divergence has taken place, and this genetic divergence leads to reproductive isolation because these rabbits look different from these rabbits. Okay? So this is an example of sympatric speciation, which means evolution of a new species from pre-existing species, but which lives in the same or occupies the same geographical area. Symbolically or diagrammatically, sympatric speciation has been shown to you. Starts with a single large population. As you can see, time is passing. Okay? Differences among individuals causes them to choose different habitats randomly. Cause is not known. Non-random matings result in two genetically distinct and separate populations that become different species. Sympatric speciation does not involve geographical separation of populations. It requires, usually, hota kya hai sympatric speciation mein, ke it requires some form of reproductive isolating mechanism which has arisen by selection within a geographically confined area. The isolating mechanism can be ecological, behavioral, structural, morphological. ये जो बहुत सारे causes अभी बताए, all different kinds of reasons due to which reproductive isolation takes place, and there is an interruption to gene flow between those two subpopulations within one geographical area. Allopatric speciation, sympatric speciation. Again, remember, both these are being described under the heading of intra-specific speciation. ठीक है? Interspecific is the same thing which we have done allopolyploidy अभी हमने किया था, right? And I was teaching you polyploidy. I told you कि auto भी होता है, allo भी होता है. And when I was teaching you allopolyploidy, I gave you this example. ठीक है, allo means one species, another different species, ठीक है, since they are different, so when they mate they produce a hybrid, and this hybrid is sterile, ठीक है. It cannot reproduce because the separation of the homologous chromosomes does not take place at meiosis, but because of random errors during mitosis or meiosis, chromosomal doubling takes place. तो अब जब chromosomal doubling आपकी हो चुकी है, so this is fertile, it is not sterile. It will be fertile अगर जो है वो हम इसको इसी की तरह की स्पीशीज से, मतलब ऐसे ही बिल्कुल emmer wheat which is AABB, if we are going to cross it with the same kind of species, it can give rise to fertile offspring. लेकिन इस क्रॉस में क्या दिखाया गया है कि अगर इसको we cross it with another species, again it will give you a sterile hybrid, and उसके बाद chromosomal doubling जो है वो हो सकती है. So this is an example of speciation, which is the result of interspecific hybridization, meaning the hybridization between plants belonging to two different species. तो ये भी speciation की एक example है, लेकिन किसकी example है? Interspecific hybridization, right? So these are just some bullet points, ठीक है? I'm just waiting for 1 minute so that you guys can go through them quickly. Okay, so we are starting with a new subtopic, which is molecular comparison of species. Okay, the name tells you, okay, we are trying to compare the species as far as their molecular structure is concerned. If you remember, when I was doing microarray with you, I told you that microarray can be used for 2 or 3 different purposes. We can do microarray to find out the gene expression between 2 cells, a normal cell and a cancer cell, and also we can use microarray to find out how much difference or how much one species differs from another species. What is their evolutionary relationship? Okay? Acha. This is the modern views on evolution. The theory of evolution has been modified in the light of genetics. For example, I would put it like this, that when biologists have to give evidence for the theory of evolution, they try to take evidence from these branches, sub branches of science or biology. So they take evidence from genetics, and they take it from molecular biology, from the study of fossils, which is paleontology, from ecology, and from ethology, which is study of behavior. So which one are we doing right now? We are going to study molecular biology, right? And in a way, genetics also. How can we compare 2 species at a molecular level? So what do? We look at some common proteins and their structures, and especially those common proteins which are very crucial to life. For example, hemoglobin and cytochrome C. Hemoglobin is a protein, you all know it's crucial to life because it is the major oxygen carrying pigment in your blood. And cytochrome C is, agar aapko yaad ho, it's a protein that is important for your cellular respiration. Agar तो इसमें कुछ मसला हो गया, so cellular respiration cannot go on. So what do we do when I want to compare 2 different species? I study the amino acid sequence, primary structure of these constitutive proteins. I hope कि constitutive का मतलब सबको आ रहा है. Constitutive का मतलब ये है कि a protein that makes up your body constitution, your body structure, जिसके बगैर आपकी जिंदगी नहीं है, right? So we've taken 2 such proteins, and one is hemoglobin and the other is cytochrome C. And then we try to see, यानी हम क्या करते हैं, different species उठा लेते हैं, and then we, for example, if I'm comparing the molecular structure of hemoglobin protein in a monkey and hemoglobin protein in human, and I do like that. So, for example, in this, they're showing you that when they studied the difference between cytochrome C structure in humans and monkeys, this was the difference of amino acids between monkeys and humans was 1, in pigs it was 10, and in fish was 21 amino acids, right? And as per the evolution theory, they say that the lesser the difference in the number of the amino acids, the more close are two species as far as their evolutionary relationship is concerned. यानी कि हम इस तरह कह सकते हैं कि for example over here it shows you a species comparison between horse and man. And कैसे compare किया? By studying the protein cytochrome C in horse and man. So it was found out that a number of the differences, the amino acid differences between the cytochrome C in the horse and cytochrome C in the man was a difference of 12 amino acids, or horse or pig में 3 amino acids है. And the divergence of the lines in millions of years, अगर आप देखें, तो it's 33 for horse and pig, and it's 130 for horse and man. क्यों ऐसे है? इसका मतलब ये है कि the lesser this number, एग्जाम में आपसे पूछा जाएगा कि the lesser is this number, the more close those two species are with each other as far as their common ancestor is concerned. This is how you can put it into words, right? Acha. This is one way in which, by means of which, we can study evolution on the basis of comparing the different molecular structures of the essential proteins or constitutive proteins. और एक और तरीका ये है कि we can analyze the nucleotide sequence of mitochondrial DNA, right? Analyze the nucleotide sequence of mitochondrial DNA. आप सबको पता है कि whether it's a plant cell or it's an animal cell, both have mitochondria. ठीक है? Let's say this is a plant cell, इसका अपना nucleus है, and it has a mitochondrion. Mitochondria has its own DNA, circular DNA. Similarly, आपके animal cell के अंदर भी mitochondria है, उसके पास भी अपना DNA है, and that is known as the mitochondrial DNA. Now the question is, why do we choose mitochondrial DNA? हम क्यों choose करते हैं mitochondrial DNA? And the reason for this is that this is the DNA that is inherited through the female line. यानी कि हम सबके पास जो mitochondria का DNA है, we have inherited it through our mother only. I'll tell you why we have inherited through our mother. In mitochondrial DNA is circular, so changes in the nucleotide sequence can occur only through mutations, because circular DNA में crossing over नहीं हो सकती. ठीक है? Crossing over के लिए it's a must के the DNA should be linear. ऐसे होती है ना crossing over. तो circular में तो नहीं हो सकती crossing over. अच्छा, so if any change is happening in the mitochondrial DNA, it is only due to mutations. Mitochondrial DNA is a naked DNA, and so no protection takes place by histone proteins, and hence it is more prone to errors by mutation. So that is why, in order to study the genetic differences between two species, we can also do that by studying the nucleotide sequence of mitochondrial DNA. Why have we chosen this? Because mitochondrial DNA is very special. It is circular, so इसमें कोई अगर change आता है तो crossing over की वजह से नहीं हो रहा होगा. It will only be due to mutations. And since mitochondrial DNA does not have histone proteins to protect it, so it's more prone to errors by mutations. So errors, if any, can be very easily picked up in the mitochondrial DNA. Different human populations show differences in the nucleotide sequence of mitochondrial DNA. So this provides evidence for the origin of different populations. Constant rate of mutation for millions of years, estimation of clock from fossil evidence. These are all assumptions. No conclusive evidence has been given for all these. All these assumptions have not been proved, as it shows, because it's just a hypothesis, right? Yes, this says that there are many assumptions which are already made. First of all, the first assumption that whatever changes are happening, they will happen at the level of the mitochondrial DNA due to mutation. And then this rate of mutation is also constant, and not only constant for 1 year, for millions of years. And if this happens in this manner, then we can actually trace the common ancestor right? And I'll tell you how. This is how your textbook explains it. You can go through this. In fact, I'll read it for you. Different human populations show differences in mitochondrial DNA sequences. These provide evidence for origin of Homo sapiens in Africa and for the subsequent migrations of species around the world. These studies have led to the suggestion that all modern humans of whatever race are descendants from one woman called mitochondrial Eve, who lived in Africa between 150,000 and 200,000 years ago. This date is derived from the molecular clock hypothesis, which I said it's an assumption. And this hypothesis assumes that this rate of mutation that is occurring in mitochondrial DNA, जो कि एक mitochondrial Eve से आया है, it is occurring at a constant rate for millions of years, right? So this assumes a constant rate of mutation over time, and that the greater the number of differences in the sequence of nucleotides in the mitochondrial DNA, the longer ago those individuals shared a common ancestor. This clock can be calibrated by comparing the nucleotide sequences of species whose date of speciation can be estimated from fossil evidence. To sum up, I could say that they say that we will study the differences in the nucleotide sequences of mitochondrial DNA of different human beings. Or jitna kam difference aayega, we would say that they are more closely related to each other as far as their common ancestor is concerned. Not only we can do that if we compare different humans, but we can do that if we compare humans and other animals also. उसमें हम एक essential protein के amino acid का sequence देख रहे थे. इसमें we are looking at the differences in the mitochondrial DNA, right? अच्छा. And again, उसमें जैसे था ना, the greater, जब हम cytochrome C देख रहे थे, तो I told you that the greater the difference in the number of the amino acid residues, the more far apart those two species are away from each other as far as their evolutionary relationship is concerned. इसी तरह से, when we study the nucleotide sequence of mitochondrial DNA between two species, so the greater the number of differences in the mitochondrial DNA, the longer ago those organisms shared a common ancestor or they are distantly related. Okay? Let me again put it in a simple manner. The greater the number of the differences in the nucleotide sequence of the mitochondrial DNA, the distantly related are those organisms from each other. यानी कि एक दूसरे के साथ उनका जो common ancestor है वो कहीं दूर पीछे generations में होगा, ठीक है, अगर number of differences ज़्यादा है. I will stop here because I will be doing an example with you after discussion.